Chapter 6 gives us an overview of what we call epigenetics and the disease process. And, and the overview here for you to take away, and again, remember my job here is to give you an idea of how to simplify your studying. So we want to talk a little bit about what we know about the study of genetics and how certain stressors impact the turning on or the turning off of genes that are already there, or by exposure to certain things, how the genes become mutated. So we know that nutrition plays a role. We don't truly understand how nutritional deprivation and the disease process over generations takes place, but we do know that certain exposure to chemicals while pregnant seem to lead to similar kinds of genetic modifications. And when we begin to talk about cancer, we know that nutrition plays a role there also. What we do know is that when the fetus is being carried in utero, there are significant factors that we've studied for years and years that negatively alter the methylation states and create a stressor that creates a genetic modification. That's why there's such a big push in today's society for prenatal care. We know that women who get prenatal care have far less serious genetic outcomes and negative outcomes because their care was monitored during that process, and exposure to those stressors were mediated through a variety of processes, taking prenatal vitamins, making sure you're not eating tuna, which carries a lot of mercury, making sure that you get genetic testing so that we know what kinds of things you can't alter. So we know that maternal care care during pregnancy greatly decreases the genetic mutations that happen to the fetus. We absolutely know without a doubt that exposure to alcohol during gestation creates a problem. The exposure in utero impacts the DNA in a negative way, creates a lot of neurological deficiencies, and that when fetuses are born and newborns are born to mothers who have continued alcohol drinking during pregnancy, they actually manifest something called fetal alcohol syndrome, which is a severe pediatric emergency. So we have learned that no alcohol during pregnancy is best for maximizing good genetic growth. Lastly, we understand that that people carry often genes that either turn on a cancer or turn off a cancer that are genetically passed along. And we know that these genes do not necessarily turn on the cancer, that there are other exposures that take place in order to make that happen. So here's where diet comes into play. We understand that high sugar, high fat diets tend to turn on these genes more so than diets that are well balanced. We are beginning to understand that exercise or the lack of exercise plays a factor in whether these genes get turned on or off. It's just an interesting thing about what we're learning about cancer. We still don't know how it happens, although we do understand by studying, say, the BRCA1 gene breast cancer women, we do understand more so, and drugs have been developed that that target specific parts of the cell growth, and we've learned that by studying genetics and how cells multiply. And this is just a nice structure of how normal cells are supposed to regulate themselves, and how a cancer cell gets, all the different areas where the cancer cell is created because of malfunctions in processes. Just an overview, not that you have to memorize it, but a really good picture of how it happens. And that's it for our overview of epigenetics. We're going to get more into cancer in the next lecture, so stay tuned.